zinc remains the most potent and the most common trace element for the promotion of the expression of methallothionein. This is an overview of methallothionein gene, and the methallothionein promoter has various response elements. We have the glucocorticoid response element, which is activated by glucocorticoid. We have some elements that are activated by start via cytokine signaling. We have the metal response element, which is activated by metal responsive transferrin factor 1 after zinc occupancies, which is a function of dietary zinc supply. Also, we have antioxidant response element, and this methallothionein is synthesized as an haploprotein. We call it thionein, but immediately it binds to metal, it becomes methallothionein. Metal ions, cytokines, growth factors, native stress radiation, all these stimulates and induce methallothionein, and the downstream effect of methallothionein on some proteins like p53 and the transcription factor NFKP influence cell survival, cell growth, drug resistance, and differentiation. Now, what is the role of methallothionein in oncogenesis? In relation to cancer, major focus has been devoted to the interaction between p53 genes and methallothionein. p53 is the tumor suppressor functions to arrest cell cycle and also to promote apoptosis. But mutation of p53 genes have been found in most human cancers, which will lead to the inactivation of p53, resulting in inactivation of p53, and thereby prevents the regulation of apoptosis. This figure will explain the role of methallothionein in oncogenesis. p53 requires zinc for its activation, maintenance, and stability, but methallothionein synthesized as an haploprotein will require zinc for its stability. So methallothionein will abstract zinc from p53, and when zinc is abstracted from p53, p53 will be inactivated, and therefore all the key components of apoptosis that p53 activates the transmission, such as caspase 1 and 3 cytochrome C leakage, they are all down regulated, which will lead to the development of cancer. Also, this methallothionein can act as a tumor suppressor. Inflammation is the body's first response to injury and infection, and inflammation is essential to healing. But when inflammation is too much, or when it goes on for a long time, it can affect the cells and destroy the cells' DNA. So methallothionein upregulates some anti-inflammatory mediators such as interleukin 10, and then down-regulates some pro-inflammatory mediators such as TNF alpha and NFKB. Methallothionein also can act as an antioxidant, because during oxidative stress, cell protects itself by using various molecules as scavengers. One of the molecules is the glutathione complex. However, the free thiol group of methallothionein can also scavenge these free radicals. Also, glutathione and methallothionein, they work synergistically, because during oxidative stress, glutathione is oxidized, and then toxic metals that are released, methallothionein binds to this metal for removal and exclusion. Also, when methallothionein is oxidized itself, zinc ions they are released, and zinc ions they can inhibit the inducible nitric oxide synthase, which will now down-regulate and lower the production of nitric oxide. In conclusion, though overexpression of methallothionein in tumor cells might have important implications in establishing neoplasm as a prognostic marker for therapy response and patient survival, data regarding the presumed role of methallothionein in neoplastic transformation are still lacking. So further studies will be needed to closely establish the dual role that methallothionein play in healthy cells and cancer cells during tumor progression and treatment. These are my references.